Hello students, in this video we are discussing about anatomy of dicot stem. Actually, in our syllabus we have secondary growth in dicot stems. But to understand secondary growth, we have to understand the anatomy of young stem. Young stem means which is a primary stem. For that, we have to take one young stem of dicot plant and we have to cut that stem in transverse manner. Means we have to take a TS of dicot stem. Okay. After transverse section, we can get a very thin section like this. Okay. This is section. Here I am drawing an outline which indicates a single layer of outer layer which is known as epidermis. Epidermis is outermost layer of stem. It gives protection for the internal tissues. Next, we can also observe few hair-like projections from the few epidermal cells. These hairs are called epidermal hairs. Since they are originating from the epidermis, these structures are called as epidermal hairs or trichomes. These trichomes are multicellular, consist many cells. Next, beneath the epidermis, we can see cortex. Cortex is differentiated into 3 zones. Now, I am drawing hypodermis, which is outer part of cortex. This portion consists collenchymatous cells. Next, I am drawing innermost cortex layer, that is endodermis. Endo means innermost. Okay, endodermis. It is also consist a single layer of cells. Okay, a single layer of barrel shaped cells. Next, the cells are closely arranged in this layer and also reserve some starch grains. That's why this layer also known as starch layer. Okay. Next, beneath to the endodermis, there will be pericycle. Consist 2 to 4 or 5 layers of cells, which is consist 2 types of cells. Sclerenchyma. Now I am drawing sclerenchyma strips, which are arranged opposite to the vascular bundles. Alternatively, there will be parenchyma tissue. Okay. Next, very opposite to this sclerenchymatous pericycle, we can see a strip of parenchyma, otherwise called as phloem. Phloem. These strips are phloem tissues. Okay. Means from here we have to consider these structures as vascular bundles. In dicots, vascular bundles are arranged in a ring-like manner, in one ring. That's why this stele is called as U-stele. Okay, this is characteristic feature of dicot stem. Now, I am drawing one more small strip, which is called as vascular cambium. It is next to the phloem. Okay. It is vascular cambium, which is useful for the secondary growth. Next, here I am drawing xylem. The xylem arranged in an endarc manner. Endarc mean the protoxylem is placed towards center or towards pith. The metaxylem is placed towards epidermis or outer side. What is protoxylem? The small vessels of xylem is called as protoxylem. The big vessels or large vessels are known as metaxylem. Protoxylem is first formed xylem vessels. Metaxylem lately formed or recently formed xylem vessels. Okay, this is vascular bundle. This is also, here we can see a single layer of vascular bundles, and the number of vascular bundles also usually more than 6 in dicot plants. Okay, they are all arranged in a ring like manner. Here we can see phloem, a strip of cambium and xylem. Here I am striking sclerenchymatous pericycle, which is very opposite to the vascular bundles. In between bundles we can see a common type of tissue, that is parenchyma tissue, which is having contact with teeth inside and with cortex outer side. Here now I am striking sclerenchyma tissue. Actually this diagram is a ground plan. Ground plan means a holistic view of transverse section. Okay, where we can see all type of structures distantly. But next we are drawing enlarged view of transverse section. In the diagram we can see all these structures very clearly. Okay, this is phloem, consists majority of living tissue. These all tissues we can see very clearly in the next diagram. This is just for understanding. Okay, this diagram usually called as ground plan. Okay, now I will label. This is epidermis, which is an outermost layer, consists one layer of cells. Okay, on the epidermis, in some plants we can see thick wall material, which is known as cuticle. Cuticle is a waxy coating on epidermis, which is useful for the prevention of water loss. This is cuticle layer. This is trichome or epidermal hair. Okay, this is hypodermis, which is outermost part in cortex, hypodermis. This is middle cortex or filling tissue. This is endodermis, which consists a single layer of cells. These three together called as cortex. Okay, here it is vascular bundle, vascular bundle, and here in the center there will be parenchymatous tissue called pith or medulla. Okay, in between vascular bundles also there will be simple parenchyma tissue called as medullary rays. These rays are directly contact with cortex. Now this is vascular bundle. Now I will maximize this for easy understanding. Okay, now I am drawing one vascular bundle. This is vascular bundle. Towards pith there will be xylem. This is xylem. This is a meristematic strip known as vascular cambium. This is phloem. Okay, this is phloem. This is xylem. In between xylem vessels, again there will be simple parenchyma, and we can also see other vascular elements like xylem fibers, xylem vessels, xylem parenchyma, all these things. This is vascular bundle. Okay, in dicots, the vascular bundle consists protoxylem towards pith, metaxylem towards epidermis or towards cortex. In between xylem and phloem, there will be cambium, known as vascular cambium. It is phloem. Okay. In dicots, we can see conjoint, collateral, and open type of vascular bundles. The vascular bundles are conjoint, collateral, and open type. Conjoint means xylem and phloem lie on same radius. Okay. Collateral means phloem placed towards outer side, xylem placed towards inside. Open means there will be cambium in between xylem and phloem. Open type of vascular bundles are characteristic features of dicots. It is very important point. Okay. Now this is ground plan of dicot stem transverse section. Okay. Now I try to enlarge a portion of transverse section. This is a small piece of transverse section. Now I will enlarge this portion so that we can clearly understand the tissue systems in transverse section. Okay, now here I am drawing an enlarged piece of transverse section. First, this is an outer layer which is a single cell layer called epidermis. The cells in this layer are compactly arranged without any internal spaces because it is outermost layer. That's why it functions like a barrier or a wall to protect internal tissues. Okay. In the epidermis, we can also see stomata, and as we discussed earlier, we can also see multiple trichomes from few cells of epidermis. And next, on epidermis, there will be thick layer of waxy coating which is called as cuticle layer. Cuticle layer is useful to prevent evaporation of water. Okay, it prevent evaporation of water. And next, the trichomes are multicellular, having number of cells like this. Okay, now I label all these spots. This is cuticle coat, this is stomata, this is trichome, this single layer is epidermis. Okay. Next, beneath to the epidermis, as like I shown here, there will be hypodermis, which is a part of cortex. Here we can see 2 to 4 layers of cells. These cells are collenchymatous. These cells are collenchymatous. The cell walls of collenchyma tissue consist pectic depositions in their cell wall corners. Okay, this is characteristic feature of collenchyma tissue. Okay, beneath to the epidermis, there will be 2 or 3 or 4 layers of collenchyma tissue. What is the function of collenchyma tissue? It provides tensile strength or mechanical support or flexibility to the young stems. Okay, since they are having pectic depositions in their corners, the cells giving mechanical strength to the plant parts. Okay, these are the layers of collenchyma. Here we can see pectic depositions. Usually we have 3 types of collenchyma tissues, but the most common type is angular collenchyma. Here I am drawing angular collenchyma. In this type, the pectic material deposit in the corners like angles or triangles. Okay, like this. And one more thing, here I am drawing a transverse section of stem. Means it is a just slice of stem. Okay, that's why only I am drawing 2D diagram here. But we have to imagine that these collenchyma cells are elongated. Whenever we cut a stem in longitudinal manner, we can better understand the structure of collenchyma cells. These collenchyma cells are somewhat elongated. Okay, in that longitudinal angles, there will be pectin depositions. But here, just I am drawing a slice of stem. That's why only we are drawing 2D picture only. But whenever we observe longitudinal section, we get a clear-cut idea about collenchyma tissue and the cell arrangement in collenchyma tissue. Okay, this is all about hypodermis, which is made up of collenchyma. And next, beneath to the hypodermis, there will be simple cortex or filling tissue. This cortex region consists simple parenchyma. Means the cells in this tissue are loosely arranged with lot of intercellular spaces. Okay, having intercellular spaces is a characteristic feature of parenchyma tissue. Okay, that's why it is a simple parenchyma tissue which is occupy major portion in the cortex region. Okay, this is parenchyma, consist more or less homogeneous cells. Okay, the main function of this parenchyma is storage of water and other food materials, but rarely they perform other functions also, like some cells consist chloroplast and perform photosynthesis. Means few parenchyma cells also behave like a photosynthetic tissue. Okay, and next, the innermost layer of cortex is known as endodermis. The endodermis always consists a single layer of cells. The cells in this layer barrel in shape. barrel in shape. Okay, they are arranged without any intercellular spaces and they consist starch granules. That's why this particular layer is called as starch layer. Starch layer. Okay, this is all about cortex. In the cortex we have collenchyma region in the outer side, which is known as hypodermis. Hypodermis. It is also called as outer cortex since it is placed in the outside of cortex. Okay, this is middle cortex or filling tissue. Filling tissue. It occupies major portion in cortex. The innermost single layer of cortex is known as endodermis, which is also known as starch layer. This is all about cortex. Consists three parts: endodermis, middle cortex, and hypodermis, all together called as cortex. Okay. Next, this portion. Okay, this portion is consist sclerenchymatous cells. This layer is called as pericycle, which is a starting layer of vascular steel. Here I am drawing only a vesicular bundle portion. That's why here I am drawing sclerenchyma tissue. Okay. Other than vesicular bundle region, we can see parenchyma tissue only. Means in the opposite to the vascular bundles, we can see sclerenchyma tissue. Alternatively, there will be parenchyma in between vascular bundle region. This is a characteristic feature of many dicot stems. Okay, usually the sclerenchyma is distributed patch-like manner. Okay, discontinuous manner. In very opposite to the vesicular bundles, there will be sclerenchyma tissue, and alternate to the vascular bundle, there will be simple parenchyma tissue. Okay, this is a sclerenchymatous tissue. Okay, and next to the this sclerenchymatous tissue, there will be one more strip of parenchyma, which is known as phloem. We know phloem is a food conducting complex tissue which is useful for the conduction of food materials. Food means either amino acids, enzymes, or sucrose or other sugar solutes. It consists different types of cells or elements like sieve tubes, companion cells, parenchyma, and phloem fibers. Okay, these all cells or components or elements are placed in this region. Okay, it is principal food conducting tissue. Major portion consists living cells, the two parenchyma cells. Here and there will be dead cells called fibers. The only cells which are dead in phloem region, they are fibers. Okay, this is phloem region. Okay, here we can see some cells having dot like structures which resembles sieve plates. Okay, now I'm drawing CU tube cells. This is sclerenchyma cell. This is also CU plate. Here it is sclerenchyma cells. Okay, this is about phloem. Okay, and here we have to show the sclerenchyma tissue with thick double line walls. It indicate the lignin depositions in cell walls. Okay, these thickenings are nothing but lignin depositions. Lignin depositions. Since here we have lignin depositions, the cells are dead. The cells are dead. The cell walls consist lignin depositions, which is very strong material, so that it is very rigid in nature. Due to this rigidity, it will useful for the mechanical support. Okay, since this sclerenchymatous pericycle consist lignin depositions, this layer is also useful for the mechanical support. Okay, next, here inner side to the phloem there will be xylem. The xylem arranged in an endarc manner. Endarc manner means the primary xylem placed towards center, the metaxylem placed towards outside, towards phloem side. Okay, this is metaxylem. Okay, this is protoxylem. Okay, and next, in between these xylem vessels there will be simple parenchymatous tissue. This is all simple parenchymatous tissue. Here we can see other xylem elements also. What are the other xylem elements? Xylem vessels, xylem parenchyma, xylem tracheids, xylem fibers. These are all present in this zone. Okay. This simple parenchyma tissue useful for the storage of some reserve materials. Okay, this is all about xylem. It includes xylem vessels, xylem tracheids, xylem parenchyma, xylem fibers, all these things. Okay, and here the parenchyma is continuous in between two vascular bundles, and here also there will be lignin depositions. That's why these cells also dead cells. Though they are dead cells, they are useful or they function as a water conducting cells in xylem. Okay, these are all xylem vessels. This is lignin deposition. Since lignin is depositing, these cells are dead at maturity. Okay, this cell which is placed towards phloem is bigger in size, called metaxylem. The cells placed towards pith side, it is smaller in size, called protoxylem. Okay, here again there will be simple parenchymatous tissue in the middle of stem, which is known as pith or medulla. which is a simple parenchyma tissue present in the center of stem in primary stems. During secondary growth, this pith will be crushed due to the accumulation of secondary xylem outside to the primary xylem. Okay, even the pith parenchyma extended in between two vascular bundles also. This parenchyma tissue is known as medullary race. Medullary race. Okay, this is pericycle, sclerenchymatous pericycle. Okay, it is sclerenchymatous. Okay, this is phloem. Phloem consists different elements. There, sieve tubes, companion cells, fibers, and parenchyma. These are the different types of phloem elements. Next, this is xylem. Xylem also consists different types of elements. They are tracheids, xylem vessels, xylem fibers, and xylem parenchyma. These are all elements of xylem. And one more thing I have forgotten here. In between xylem and phloem, there will be meristematic strip, which is called as vascular cambium. The cells of this vascular cambium having prominent nucleus, which is very actively dividing tissue. This is vascular cambium. Cambium or meristem both are same. Since it is present inside the vascular bundle, it is known as vascular cambium. Next, it is pith or medulla. Okay, and the parenchyma present in between vascular bundles also. But here the parenchyma is radially elongated in this manner. Okay, these parenchyma is known as race of medulla or medullary race. In the medullary race, the parenchyma cells are elongated radially due to the crushing by vascular bundles. Okay, this is a portion of enlarged part of Dicot stem. Okay, here this portion. Now firstly I will explain it once again. This is a single layered epidermis. On epidermis there will be cuticle and here and there stomata, and also we can see few trichomes which are multicellular, means they consist number of cells. This portion is cortex, which is subdivided into three regions: outer cortex, middle cortex. The middle cortex consists loosely arranged cells, and the innermost layer is endodermis. Okay, these three layers together called as cortex. Okay, next this is pericycle, consists sclerenchymatous tissue. Okay, next this is phloem, which consists majority of living tissue. Okay, these are the elements: sieve tubes, companion cells, fibers. This is parenchyma. Okay, next, this is vascular cambium. This is xylem. Consist different elements. They are tracheids, vessels, fibers, parenchyma. This is pith or medulla. It is nothing but parenchyma. Okay, this is all about anatomy of dicot stem. Okay, thank you.